Hi everyone, thanks for joining me for this video today. My name's Adrian Shembry. I'm a Director of Clinical Science at Cogstate. I'm based in our Melbourne office in Australia. I'm a clinical psychologist by background, and today I'll be presenting this video on designing computerized test batteries for clinical trials. So throughout this video, we'll start off by going through some of the factors that should be considered when selecting a cognitive test battery. So for example, what are the ins and outs that we really need to be thinking about when trying to select the optimal tests for a given study? Following this, I'll present 3 case study publications that were recently published on the Cogstate computerized test battery being used in several different contexts. Firstly, we'll have a look at a study that used a safety monitoring battery administered to healthy adults in context of a novel antidepressant being administered. In the second study, we'll have a look at an Alzheimer's disease study that used a large neurocognitive test battery as an exploratory efficacy endpoint. And then finally, we'll discuss a schizophrenia study which also looked at efficacy using a set of test studies often used in the context of schizophrenia. So to begin with, let's have a talk about some of the ins and outs of selecting a cognitive test battery and what are some of the important considerations that we need to think about. One of these is the context. So what is the context in terms of how the battery is going to be used? Is it going to be used in a study where there are multiple assessments within a brief time period, or are the assessments more spread out? Are the assessments going to be administered all in clinic under supervision? Or is it possible that some of them might be remote? And in thinking about remote, will they be remote supervised assessments or will they be remote unsupervised assessments? So these are some of the contexts that we need to think about when selecting optimal tests. Secondly, the disease. What are the cognitive effects associated with the disease being studied, and are they known? Are there previous studies that have used either computerized or pencil and paper tests where certain cognitive effects have been documented? This can give us clues in terms of the ideal cognitive domains that we might want to incorporate into a cognitive test battery. An example of this is, in terms of concussion and episodes of concussion in sport. What we often see is that following a concussion, an athlete will undergo a slowing in terms of reaction time or processing speed cognitive measures. And so when we're thinking about test batteries in the context of concussion, we often like to include several tests that measure processing speed or reaction time. Reaction time is also a cognitive domain that is impacted in diseases such as depression. So in major depressive disorder, we often see that there is a slowing in processing speed or reaction time as a result of the condition itself. And so when we're looking at treatments for depression, this is often a cognitive domain that we measure. By contrast, when we're thinking about neurodegenerative disorders such as Alzheimer's disease, typically reaction time tends to be relatively well intact. So the focus in those contexts is more on aspects such as memory. Another area that we need to think about is the drug itself or the treatment. So what is the underlying mechanism of action for the treatment under investigation? And this again can be based on, what is the area of the brain that is likely to be impacted, or are there previous studies that we know of where we have seen certain deficits or improvements in particular cognitive domains? If these are known, then it can steer us in a direction of ensuring we include certain tests of those underlying domains. And lastly, we need to think about subject level factors. So are there particular factors of this study population that we need to take into consideration when selecting the cognitive test battery? For example, what is the age of the population? Are they pediatrics? Are they children? And are they adolescents or adults? If they are adults, are they older adults? The nature of the demographic characteristics can help us to decide whether we're selecting a battery that is too difficult, whether the subject population will be able to complete all of the tests, and also whether they'll be able to tolerate a lengthier battery. So we need to think about patient burden. And when we're thinking about batteries for people with particular diseases of a given severity, or when we're thinking about test batteries for children, we need to think about minimizing patient burden in terms of the length of the battery. So these are all characteristics that we need to think about when selecting an optimal cognitive battery. All right, so let's jump in and have a look at some of the publications that I was talking about earlier. The first one is actually a safety monitoring study that investigated an investigational drug, which was an antidepressant, in a healthy adult population. Now, the primary aim of this study was to evaluate the effect of intranasal esketamine on cognition when administered at an 84 milligram dose, and the duration of any cognitive effects as assessed using a computerized test battery. Now, a secondary aim was to evaluate sleepiness and also the safety and tolerability of intranasal esketamine. Now, for anyone unfamiliar, ketamine is an NMDA receptor antagonist that exerts a rapid onset of antidepressant in patients with treatment-resistant depression. Now, esketamine is 3 to 4 times more potent than ketamine at the NMDA receptor site, enabling antidepressant effects at lower doses and with potentially fewer side effects. Now, esketamine nasal spray was actually recently approved by the US Food and Drug Administration for treatment-resistant depression, so it is certainly an option that is showing a lot of promise. So let's dive in and have a look at the study design. We can see that 24 subjects were screened and randomized, and half of these were randomized to treatment sequence 1 and half were randomized to treatment sequence 2. So for those in treatment sequence 1, they were first administered intranasal esketamine, followed by an intranasal placebo in period 2. By contrast, treatment sequence 2 was administered intranasal placebo in period 1, followed by intranasal esketamine in period 2, and there was a 7-day washout in between the two periods. Here's a summary table of the study participants, and we can see here the demographic characteristics across treatment sequence 1 and 2. What we can see is that the mean age of participants was very similar across the two groups, that there was an equal proportion of males and females within the groups, that the majority of people in the study were white, and also at baseline, mean BMI was within the healthy weight range, so it was within that 20 to 25 range. Here's a summary of the cognitive test battery that was used in this particular study, and we can see that it incorporated 5 tests from the Cogstate computerized test battery, those being the detection and identification tests, which are 2 measures of processing speed, where we measure reaction time as the primary outcome. Then we have 2 memory tests, one being one-card learning, which is a visual learning test that uses a pattern separation paradigm. And then we have one-back, which is actually using an n-back paradigm to measure working memory. The last test in this battery was the Groton-Maze learning test. Now, this is an executive function test that uses a hidden maze paradigm. And importantly, even though this was a study in healthy adults, several of these tests could be used in future studies within a patient population in depression. So as I mentioned earlier, psychomotor functioning or reaction time is often implicated in depression, as is executive function and memory. And therefore, even though this study was in healthy adults, this study would provide useful information in the context of future studies within a clinical population. Now, the schedule of assessments in this study was quite condensed. So there was a baseline assessment 1 hour prior to dosing, and then follow-up assessments at 40 minutes, 2 hours, 4 hours, and 6 hours. So given that the cognitive test assessments were 1 to 2 hours apart, the study team would have had to be really conscious of the length of the battery, given that other assessments would have been fitting in between as well. Let's have a look at the safety profile that emerged from this study. So we can see here a summary table of the treatment emergent adverse events. What we can see here is that the incidence of these in the intranasal esketamine group was considerably higher. In fact, 100% of subjects in that group experienced 1 or more TEAE, when compared with placebo, who experienced 37.5% of subjects in that group had one of these events occur. Now, the most frequently occurring event was dizziness, and that occurred for 16 of the 24 subjects in the intranasal esketamine group. Now, importantly, all treatment emergent adverse events did resolve at the end of the study. When we look at the cognitive safety profile for this study, some really interesting results emerge. What we can see is that across these 4 tests in the battery, here we're looking at the detection, identification, one-card learning in the bottom left, and one-back in the bottom right. And what we can see is that the blue line, which represents intranasal placebo, and then the green line, which represents intranasal esketamine, they follow quite similar trends with a minor difference. So we can see that the blue lines are relatively flat across the assessments. So the placebo doesn't seem to be having an effect on cognition. However, when we look at intranasal esketamine, we can see that it's quite similar to placebo at baseline, but there is a decline evident across each of these tests at the 40-minute post-dose assessment. So we can see, for example, on the detection and identification tests, performance is worsening as demonstrated by an increase in performance in the green line, indicating a slowing of reaction time. Now this then returns to baseline levels by the 2-hour assessment and remains relatively flat at the 4- and 6-hour assessments. When we look at one-card learning in the bottom left graph, we can see that this is a measure of accuracy. We can see that accuracy declines at 40 minutes relative to baseline and then returns to baseline levels after that. Likewise, with one-back, which is another measure of reaction time, we can see slowing at 40 minutes relative to baseline. So we can see a lot of the cognitive effects in this study occurred at the 40-minute time point and then returned to baseline shortly after. This was even more so pronounced for the Groton-Mays learning test, which, as I mentioned earlier, is a measure of executive function. We can see relatively flat performance for the intranasal placebo group. However, for the esketamine group, we can see more errors being made on this test at 40 minutes post-dose relative to the baseline assessment, and then again, a return to baseline at 2 hours and onwards. So the results here indicated that a single dose of intranasal esketamine was associated with decline in cognitive function compared with placebo, but this was specific to the 40 minutes post-dose assessment. Importantly, cognition did return to comparable levels by 2 hours post-dose, and there were no significant differences between placebo and the intranasal esketamine group observed at the 2, 4, or 6-hour time points for any of the 5 cognitive tests in the battery. Now, the effect of intranasal esketamine on increased sleepiness was also measured, and it was actually more sustained relative to the effects of cognition. So increased sleepiness was experienced and didn't return to near baseline levels until 4 hours post-dose. So the effect on cognition was resolved by 2 hours, the effect on sleepiness resolved by 4 hours. So the study team concluded that a single intranasal dose of esketamine, 84 milligrams, was safe and well tolerated in healthy participants with no new safety findings of clinical concern. Let's have a talk about the second study. Now, this study was actually a study in Alzheimer's disease and was actually looking at safety, tolerability, and efficacy. So the study was conducted on the investigational drug PQ912. Now, prior to this study, this drug had undergone considerable preclinical and clinical investigations, including both in vivo animal and phase 1 studies, and it was considered to be a first-in-class QC inhibitor in clinical development. Now, this was a study that was published by Sheltons and colleagues, published in 2018, and it was a phase 2 study, phase 2A study, with the objective to investigate the highest dose of PQ912 that was used in an earlier phase 1 study. And it was also aimed to evaluate safety and tolerability signals in subjects with MCI due to Alzheimer's disease or mild dementia due to Alzheimer's disease after 12 weeks of treatment. Now, a secondary aim was to evaluate the usefulness of various efficacy measurements for future studies that would use longer treatment durations. And this is an important point because the cognitive test battery in this study was actually used as an exploratory efficacy endpoint. And even though the treatment duration in this study was only 12 weeks, there was potential for future studies to have much broader and longer treatment durations. So this study would give some indications of possible cognitive effects within the short term of 12 weeks. When we have a look at the study design of this study, we can see that 238 subjects were assessed for eligibility, of which 118 were excluded, and that left 120 that were randomized to participate, 60 of which were allocated to a PQ912 group and 60 were allocated to placebo. We can see that none of the subjects in either group were lost to follow-up. However, in the PQ912 group, 5 did discontinue because they withdrew consent, and a further 16 discontinued due to adverse events. There were no discontinuations within the placebo group. Here we have a summary table, again, of the study participants. We can see that the age distribution was quite similar for both groups, ranging between 70 and 72 years. Importantly, there were slightly more women in the PQ912 group, so slightly more females in that group compared with placebo. So placebo was closer to 50% male and female, whereas PQ912 was slightly more females. Importantly, carriage of ApoE4 allele was as expected, and it was equivalent across the two study groups. So 63% in the PQ912 group and 72% in the placebo group. Here's a summary of the cognitive test battery that was used in this study. We can see that there were 7 tests overall, but interestingly, 5 of these were Cogstate computerized tests and 2 of these were pencil and paper tests. So we can see that the 7 tests were actually clustered into 3 composite scores. The first being an attention composite comprising the detection and identification tests. Then we have an episodic memory composite. The first test being one-card learning, which, as I mentioned earlier, is a visual memory test which uses a pattern separation paradigm. The second memory test, however, was the International Shopping List test, which is actually a verbal list learning test. So the 2 memory tests are actually going about assessing memory in quite a different way. Finally, we have an executive function test, an executive function composite, which incorporated the one-back test, which is a measure of working memory, and then we have 2 pencil and paper tests, so the letter fluency test and category fluency. So for anyone unfamiliar, letter fluency is where the subject has to say and think of as many words as they can, beginning with a certain letter within a predefined period of time. Category fluency is where they have to say as many words of a certain category that they can, again, within a pre-specified amount of time. So this test battery was fairly broad, and in addition to these composites, all tests did combine into an overall cognition composite. Again, let's have a look at the treatment emergent adverse events profile for this study. Here we compare placebo with PQ912. And of note, there was an approximately equal total number of subjects with TEAEs across the two groups. However, 20 subjects from the PQ912 group actually discontinued compared to none in the placebo group. Now, it's also noteworthy that the TEAEs tended to be in the moderate to severe range in the PQ912 group, and they were generally mild for the placebo group. So this suggests that whilst the total number of subjects impacted was quite similar, the nature of the treatment emergent adverse events was quite different, whereby there were more serious adverse events in the PQ912 group and more serious TEAEs in the PQ912 group when compared with placebo. The efficacy findings in this study were actually quite interesting, because a very interesting and exciting finding from this study was that the pharmacokinetic measures suggested that the target was engaged. Now also, the patients in the study showed no adverse clinical outcomes, and from a cognitive perspective, there was no decline, which is very important in the context of an Alzheimer's disease study. Now, what was also very interesting, and we can see it in the 3rd row of the summary table at the moment, is that there was actually a treatment effect observed for the one-back test, and this was within the intention to treat population. Now, in this population, we can see that reaction times were slightly faster in the PQ912 group relative to placebo. Now, a treatment effect of 0.21, so that was an effect size, a Cohen's d of 0.21, was also found for the detection test. However, it was not significant. Overall, there were no significant treatment effects found for the episodic memory, executive function, attention, or overall cognition composites. And there was also no difference in MMSE scores across the two groups. There were also no effects on gender, ApoE, carrier status, country, or age. So this is an important result because it suggests that even though there wasn't improvement in performance in the PQ912 group, other than the one-back test, which is quite an interesting finding, there was also no decline. And the study team concluded that the exploratory efficacy findings in this study are promising, as the early signals found all are pointing in the same direction of improving function and inhibiting deterioration of synaptic activity and reducing neuroinflammation. I want to come back to that finding in terms of the one-back test, because it really was an interesting result. And the study team did dive a little bit deeper in terms of interpreting this particular finding. Now, they did so by looking at baseline levels of cognitive impairment. And we can see in the summary plot on the left on screen at the moment that the greatest deficits at baseline were actually on the ISLT, both the immediate and delayed recall components of this test, with impairment being somewhere between the 2 and 3 standard deviations below normal. When we look at the one-back test, we can see that on average, the deficit was about -0.7, which indicates that it's heading towards the mild impairment range. Now, in this context, the study team concluded that an improvement of a 0.23 effect size is actually in the direction of a substantial return toward normal function, and that this might indicate early beneficial changes of PQ912 on cognition due to a more acute rescue effect on impaired synapses. So quite an exciting study, that one. Okay, the third study that I'd like to discuss today was also looking at efficacy. However, it was looking at efficacy in the context of schizophrenia. So this study included an initial acute treatment period that was placebo-controlled, with subjects randomized to 2 fixed doses of lorazepam and also an active control, which was quetiapine XR given at an approved dose. Now, the inclusion of a placebo in the acute phase allowed for an assessment of the extent to which practice effects may contribute to improvement in cognition. Now, the study team then conducted a double-blind extension phase, which allowed for examination of cognitive treatment effects for up to 6 months after the acute study period. Now, this was the first study to evaluate both short and longer term effects of lorazepam on cognitive performance in schizophrenia, where there was a double-blind design, but also both active and placebo controls. Okay, here we can see a summary table of the study design. Overall, there were 488 subjects randomized, so it was quite a large study, and they were randomized to 4 groups during the acute phase, and there was an fairly equal proportion of subjects across these 4 groups. There was some dropout during the acute phase, and overall, we can see that between 61% and 81% completed this particular phase. There was then a Cogstate computerized battery completed at the end of the acute phase and also incorporated during the extension phase. When we have a look at the demographic characteristics across this particular study, we can see that there is a similar age distribution during the 4 groups during the acute phase, but there is also similar age distribution across the 3 groups in the extension phase. We have more males than females in each of the groups here, so that's fairly typical of schizophrenia studies. And also when we look at PANSS total, so the positive and negative symptom scale total scores at baseline, we can see very similar levels of performance across the different groups. Importantly, at the bottom of the table here, we can see pre-treatment Cogstate composite Z-score distributions, and we can see that for the full analysis sample, typically the average deficit was between 3 and 4.5 standard deviations below normal, and for the evaluable sample, this was typically between 2.5 and 3.5 standard deviations below normal. So quite a strong level of impairment in this particular study. When we look at the cognitive test battery that was used, we can see that there were 7 tests that were used in the Cogstate computerized test battery. These incorporated several tests that we've talked about so far today, including detection and identification, both measures of processing speed. We had one-card learning and one-back. Also the international shopping list test, which are the 3 measures of memory. And then we had Groton-Mays learning, which, as I mentioned earlier, is a measure of executive function. And the 7th test in this study was the social emotional cognition test, which is a measure of social emotional cognition, whereby the subject sees 4 faces on the screen and they have to pick the face which is showing a different emotion to the other 3 faces on the screen. So it's using an odd one out paradigm. Now, in this study, the schedule of assessments had a pre-treatment baseline and then a week 6 assessment, which was actually an end of acute study phase assessment. There were then 2 assessments during the extension phase, which were at weeks 19 and 32, and this corresponded to the months 3 and 6 time points. Now, the primary outcome in this study was actually performance on this particular composite. So it was an entire composite comprising all 7 tests, and that was computed by taking an average of the standardized Z-scores across the 7 tests. Now, if 3 or more tests were missing at a particular visit, the composite was just set to missing overall. So let's have a look at some of the study findings. What we can see was in this study, based on pre-specified ANCOVA models, there were no significant differences between the groups for the full analysis sample. However, when the analysis was limited to the evaluable sample, some interesting results did emerge. Specifically, there was change from baseline, which was significant for the lorazepam 160 milligram group when compared to the active control, which was the quetiapine XR group. Now, this significant difference between lorazepam 160 milligram was also evident between that group and the placebo group. When we look at the extension phase, we can see some more interesting results. Specifically, there was a change in neurocognitive function from pre-treatment baseline for the overall lorazepam group when compared with overall quetiapine XR group at both weeks 19 and 32. So these were for the groups that continued on using lorazepam 160 milligrams and the quetiapine XR group, which continued using quetiapine XR during the extension phase. Now, this important finding was evident for both the full sample and the evaluable sample. Now, the study team also went a step further and in the extension phase had a look at the impact and relationship between cognition and also functional capacity. So we can see in the plot on the left that there was a significant association between functional capacity as measured by the UPSA-B and the cognitive composite at the baseline assessment. So the second plot on the right looks at the overall longitudinal association between cognitive change and change in performance on the cognitive test battery and changes in functional capacity. We can see that these relationships were also significant and that they were also similar in terms of their trend and direction. So what this suggests is that as performance on the computerized cognitive test battery is improving, as too is functional capacity on the UPSA-B. Now, the study team concluded that, to our knowledge, this is the first study to date where an investigational drug treatment was shown to be superior to placebo for cognitive performance based on valid cognitive test scores and a functional co-primary measure at 6-week endpoint, as well as demonstrating superiority to an active comparator over an initial 6-week acute phase and a subsequent 6-month extension study period. All right. So that concludes the walkthrough of those 3 papers that did use 3 quite different cognitive test batteries, but for different contexts. So we did see some overlapping tests across the studies, but also some different rationales as to why those tests were used. So to summarize, importantly, the selection of cognitive test batteries should not be only based on the therapeutic area being studied. What it needs to do is also consider the study context, disease severity, aspects like the drug under investigation, and also any subject level factors that may be relevant to a particular study. The Cogstate computerized battery can be configured with flexibility. So we've seen many different tests in these studies that we've discussed today. And importantly, the tests used in those studies doesn't need to be the same for each time point within a study period. So it certainly can be consistent across time points. However, if a study team has more time for administration, say at a baseline and a certain follow-up assessment, we can certainly incorporate more tests at those time points and fewer tests at the assessments in between. Now, batteries should be tailored around the primary, secondary, and exploratory aims of a study. So at times that will incorporate safety, at other times it will incorporate efficacy. And as we've seen from a couple of the studies today, at times both will be looked at. And certainly a cognitive test battery can be incorporated in that context. And finally, the Cogstate science team is available to discuss any upcoming studies that you have and also cognitive test batteries that might be appropriate for those studies. We do have a broad range of experience in working across a range of therapeutic areas, and we'd be more than happy to speak with you about any upcoming studies that you might have in the pipeline. All right, thanks very much for joining me today.